So in the previous studies, we worked with some samples obtained from 10 kilometers exclusion zone of Chernobyl nuclear power plant, and we find some interesting bacteria. In the previous studies, we identified that in a genus of Pantoea, and we find that it had some greater level of UV tolerance, and you can see the strain Pantoea H7. It had a great tolerance of UV light, and it had some interesting level of fatty acids as well. So we try to identify that with some biochemical methods, like isolated DNA from culture with quick DNA plus kit, and we find some gene fragment with some primers, and we contain variable reagents V1, V2, V3, and V4 of the genes, and we purified DNA and sequence it with the Sanger method. And after that, we try to make a sequence alignment with BLAST10, and we use muscle for making phylogenetic tree, and we get some results on figure 1 as well. The position of our strain is the most likely Pantonea agglomerans, but we can't say it with 100% because we get a result that a very high level of identity. You can see on the table number 2, it's here, and we need to make some more. We need to make some more we need to make some more, because we get a very low level of difference. You can see it's very similar, but we have very similar with Pantonea eucalypti and Pantonea anatis. It's lower than 1% as well. So we need to make some more analysis as well. And in conclusion, I want to say that the 16S gene sequences confirmed for this UV tolerant strain H7 belongs to genus Pantonea, but we need, and it's like to Pantonea agglomerans. But for further special identification analysis using other, we need to use other gene marker as well. GRB is required, as 16S RNA gene was shown to share extremely high degree of similarity with another bacteria from our from our work. Maybe that's all. I'm sorry.
Thank you so much. Don't be afraid. So we're here in the friendly atmosphere, and everything was okay and perfect.
Can you repeat? Was not very good.
Yes, sometimes it was. So we just now listen up Ilya report. So we have questions. Expert committee participants. Tell me please how this bacteria, what features they provide the ultraviolet defense. So how bacteria can survive from ultraviolet?
We are seeing, you can see the results of chromatographic analysis of lipids. We are thinking that at different levels of saturated acids give for this bacteria some tolerance to UV, but it can be a very complex factor. But one of our hypotheses is that great levels of non-even chain saturated fatty acids give for this bacteria great level of tolerance.
Thank you. So it depends from the composition of the liquid. Okay, Professor Larissa Sivka also have a question.
Please tell us, what was the general purpose of your work? Why did you study this bacteria? For the practical use, for the widening our knowledge concerning biodiversity?
Is identify this strain for using it, for giving this strain to collections, and also it can be used in some kind of using that for some kind of biodiversity or biotechnology, because it's very UV tolerant bacteria as well.
Maybe for bioremediation.
Yeah. I'm sorry. Yep.
Okay, thank you so much, Ilya. You also can stop to share